Assalamu alaikum, guys. Welcome again. So, we are going to continue from where we ended up last time. We were studying about metabolism of the nucleotides. So, I'm going to share the presentation with you again. So, what we studied last time was that for the metabolic pathways of nucleotide, there were two different types of the pathways that exist. One is called as the de novo synthesis, which starts from the very basic precursor molecule. And then there's another one that is being called as salvage pathway, uh, which we have to study later on. So we have finished de novo synthesis of the nucleotide in our last class. And now I'm going to continue with the regulation of the de novo synthesis. As you are aware of the fact that all metabolic pathways are strictly controlled and regulated. And this is one of the characteristic of all metabolic pathways. And it is essentially needed as well, because if the metabolic pathways are not under the stringent control and regulation, then there will be a mere wastage of metabolites and energy. So every pathway is regulated depending upon the requirements of the cell, and that's why every pathway regulation is very important. So for the de novo synthesis, there is a significance regulation, and this regulation is number one, to meet the need of the body without wasting, as I said. Like if we need nucleotides, we synthesize them. And if we don't need, definitely we don't synthesize them. So that's how the first significance come into action. And the second one is that we have AMP and GMP control for the de novo synthesis in their respective form. If you remember that IMP was the first precursor molecule in the de novo synthesis of nucleotides. So we can say that this is the control point. This IMP is the control point for the de novo synthesis regulation. And it is controlled by the ratio of AMP, GMP, which are adenine and guanine monophosphate, versus their triphosphates, which is GTP and ATP. So, because we know that the ratio of AMP and ATP, or GMP or GTP, is an indicator of energy status of the cell as well. So that's how we have the main regulation of this de novo synthesis of nucleotides occur at the level of IMP, inosine monophosphate. So this is a pictorial representation which will give you an idea that how the regulation takes place. We go sequentially. Now, starting from this one, if you remember, the ribose 5-phosphate is being converted into PRPP by the enzyme ribose 5-phosphate pyrophosphokinases. Then this PRPP will be transformed into phosphoribosyl 1 amine by the enzyme this amidotransferase, and then finally this is by a couple of steps transformed into IMP. So IMP was the precursor molecule, if you remember. From there, it can either go towards xanthine monophosphate, and then guanine monophosphate, then di, and then triphosphate. Or alternatively, this IMP can be transformed into adenosuccinate, and then from that to AMP, that ADP and ATP. So that was the key step of the metabolism in the de novo synthesis we studied. Now, definitely the regulations are primarily done by the enzymes. Now you can see that if we have higher amount of GDP in the cell, this higher amount of the GDP is in turn affecting these two enzymes, this one and this one. So this is what we call the feedback control, because this is the, you can say that's the product of this metabolic step. So if you got the product already there, that's an indicator that you don't need more of it. So when you have a higher amount of the GDP, and ADP, which is another product, you can see that this is called the feedback control, where the product itself is inhibiting these two key enzymes of the pathway. Similarly, ADP is going to inhibit these two key enzymes of this route. So that means this is being called as feedback inhibition. And this feedback inhibition itself replicates that how the process needs to be regulated. In this case here, if you look at this one, this precursor alpha PRPP, this is an activator. Like these two, GDP and ADP, they're gonna inhibit the pathway, while the formation of this PRPP is going to enhance the pathway because that's a precursor. So when you've got more precursor, then it promotes towards more synthesis. And we can see that this precursor PRPP, if it's accumulated or it's got a higher concentration, it enhances, it triggers the reaction by activating this enzyme, PRPP aminotransferase, hence producing more of the product. So now I think from this picture, it's pretty much clear that the regulation have got two important aspect. The first thing is that when you got more of the product, there will be a feedback inhibition as shown for the GDP and ADP. And if you've got more of the precursors or intermediate, which is in this case is PRPP, it will regulate in a positive direction to have more of the product formation. So that's how the regulation takes place. So this is the pictorial representation of purine nucleotide biosynthesis, which is primarily regulated by feedback inhibition. Now, today we're going to start the second of the pathway for nucleotide biosynthesis, and that is being called as salvage pathway. I already have given you a little bit of idea in my last lecture regarding the difference of de novo synthesis and salvage pathway. So in this salvage pathway, the purine bases are created not from the scratch like in the de novo synthesis, rather they are created by degradation of RNA or DNA or their intermediates, and those degradative products of the RNA or the DNA or the degradative products of the purine can be utilized directly to convert the corresponding nucleotide. So it's not starting from the scratch. It's not starting from the basic one. It is starting from by utilizing the intermediates available, which are either produced by degradation of RNA or DNA or intermediate of the protein purine synthesis, and they can be directly utilized to synthesize uh, the corresponding nucleotide which we need. So the significance of the salvage pathway obviously is very much clear. Number one, it will save the fuel because you're not starting from the beginning. You are starting from the middle ground where you've got some of the intermediates already produced, so you don't need them to synthesize right from the beginning. So you are actually saving the fuel. It's energy efficient. And secondly, some tissues and organs like brain and bone marrow, these are only capable of synthesizing the nucleotide by salvage pathway. They lack the process of de novo synthesis. And the most common answer or the reason that why they lack is that they don't have that enzymes which actually are needed to synthesize the nucleotides right from the beginning in the de novo synthesis. So they don't have that enzymes, so they rely on only the salvage pathway to synthesize the nucleotides or biosynthesis of the nucleotides. So in this case, two phosphoribosyl transferases are involved in salvage pathway. Two of the key, I would say, enzymes which are in the category of phosphoribosyl transferases enzyme are the key enzymes involved in salvage pathway. The first of that is called APRT, which is adenine phosphoribosyl transferase, used for adenine. and HGPRT, which is hypoxanthine guanine phosphoribosyl transferase, which is primarily used for guanine or hypoxanthine. So let's start with the purine salvage pathway. Now, in the purine salvage pathway, you can see, as I mentioned earlier on, that this salvage pathway is not starting from the beginning. It's actually using the precursors or the intermediates of different catabolic pathways. Now, adenine is being converted to adenine monophosphate. This is just a transfer of a phosphate group to that. And the donor of the phosphate group is PRPP. The enzyme is adenine phosphoribosyl transferase, and you got AMP. In other case, the second enzyme which I mentioned was HGPRT. The donor of the phosphate group is the same, that's PRPP. And in this case, it's hypoxanthine or guanine that will be adding up with a phosphate group to form IMP and GMP. So it's just both of these reactions are actually transfer of the phosphate group, which is being shown here. And this is about the purine nucleotides. So adenine, hypoxanthine, and guanine. One of the clinical conditions which is mentioned here is that in some people, the HGPRT enzymes activity is not there, and that leads to a clinical condition called Leishman syndrome. I'm not going in the detail of this, just to remind you of this clinical condition, which is because of the absence of the activity of this enzyme. This syndrome was first identified or described in 1964 by two scientists, Lesh and Nahan. And as I said, that this is because of the defect, or you can say the lack of this particular enzyme HGPRT. Uh, this is more of a sex-linked metabolic disorder, which is only in the males, and the rate of the purine synthesis increased about 200 folds because of the absence of this enzyme. The loss of HGPRT leads to elevated level of the PRPP. I think that's pretty much a common sense base, because PRPP is the donor of the phosphate group that you have studied in the previous reaction. So when this PRPP is not donating the phosphate group because of the absence of that particular enzyme, then you have got more level of the PRPP or elevated level of the PRPP. And that will also trigger the de novo purine synthesis, because salvage pathway is not going on, so it triggers the de novo synthesis. And the uric acid level also rises because of the increased purine synthesis, and that results in a condition called gout. And in addition to that, there are some mental aberration and other clinical conditions. This is the Leishman syndrome, some of the pictures. Okay, the next step is the formation of the deoxyribonucleotides. So formation of the deoxyribonucleotide will involve reduction of the sugar, because we are converting the ribose to deoxyribose. So primarily in this step, we are doing the reduction of the sugar moiety. And this sugar moiety will be of ADP, GDP, CDP, or the UDP. So deoxyribonucleotide synthesis is at the level of nucleoside diphosphate NDP. Here is the reaction. You can see the deoxyribonucleotide synthesis at the NDP level. You can see that you have got the enzyme ribonucleotide reductase, because that's a reduction reaction. And you can see that this is the 5 carbon sugar. We have hydroxyl group there already. We have 2 phosphate attached. We have a nitrogenous base. The enzyme is reductase. Water goes out. Magnesium is a cofactor for this reaction. And as a result of this, you can see that it is converted to hydrogen. One of the oxygen has been removed, right? And similarly, this DNDP will be then converted to DNTP by having another phosphate group from the ATP. Now, in reverse, this NADPH which is being converted to NADP positive by using an FAD, because FAD is going to accept the hydrogen which is lost by NADPH, and that results in the formation of thioredoxin. So this is how the whole summary of how the reduction takes place from ribose to deoxyribose, and that's why we called it that this deoxyribonucleotide synthesis is going on at NDP level. So this is what the NDP stands for. So in this case, I would just summarize that 5 carbon sugar is undergoing the reductase activity, water is being lost, then the DNDP is transformed into DNTP, which is regenerated and can be utilized again, while this thyrodoxin formed here is being converted to thyrodoxin here by removing these sulfurs, and NADPH will be a donor of hydrogen converted to NADP positive, and FAD will be the acceptor. You know that in hydrogen transformation, when one of the molecule is losing electron or hydrogen, there should be an acceptor of the hydrogen as well. So that's how the reactions are coupled. Now the next thing is to discuss about anti-metabolites of purine nucleotide. Anti-metabolites of the protein purine nucleotides are actually the structural analogs of the purine. Now, what is an analog? An analog is a compound which is structurally pretty much similar, but it's not the actual compound. That's why we call them anti-metabolites, because they are not purine nucleotides, but they are a structural analog of them. So the structural analogs of the purine amino acids and folic acids are being called as anti-metabolites. What they can do, they can interfere or they can inhibit or sometimes can block the synthesis of the pathway, because they're not the actual molecule. As I said, they are kind of ghost molecule which have the structural resemblance with the original molecule, but they do not actually behave like that. So these anti-metabolites, they sometimes affect, interfere, inhibit, or totally block the synthesis of the purine nucleotides. And further, when you have purine nucleotide synthesis blocked, it will affect the synthesis of the DNA and RNA and proteins as well. So these anti-metabolites are one of the very important tool used in cancer control, because what's going on in cancer is abnormal cell multiplication. Abnormal cell multiplication means abnormal formation or excessive formation of DNA and RNA. For the DNA and RNA, you need nucleotides. So these anti-metabolites, they inhibit the synthesis of the nucleotide, thereby they inhibit the synthesis of the DNA and RNA, and thereby they inhibit the cell multiplication. And that's what the problem in cancer is. So that's why these anti-metabolites are uh, widely used for the cancer treatment. Now, there are certain purine analogs which we just mentioned in the last slide. Now, there are some specific examples. You can see 6-mercaptopurine is an analog of hypoxanthine. So this is the actual molecule, and this is the analog. And you can see it's pretty much similar with just difference of this SH group. So that's how these are anti-metabolites, as I said. This is how it's going to affect as well. The 6-MP nucleotide is an analog of IMP as well. So you can see that 6-MP is transformed in by amidotransferase, normally in the de novo synthesis, and HGPRT in case of the salvage pathway. So when you have got the 6-MP, it's going to block the entire process, and you won't get the IMP, and you won't get the final products, which is AMP and GMP. So in this case, you can see that this analog is not only affecting the salvage pathway, but it's also affecting the de novo synthesis as well. There is another example which is of amino acid analog, and that is azairacine. Why amino acid analog? Because if you remember the de novo synthesis, there were different amino acids, and their role was to give carbon and nitrogen during the de novo synthesis. So their analog are also kind of inhibitors. So you can see there this azathioprine is an analog of glutamine, and if this analog is there, it's going to impair or inhibit the role of this amino acid. Then we have another analog, and this analog is of the folic acid. Remember, the folic acid was used as a one carbon donor in de novo synthesis. So this aminopterin, AP, and methotrexate are the structural analog of folic acid. So to make it short, these structural analogs जो कि आपके साथ मैंने यहाँ तीन डिस्कस किए, one is of the folic acid, one is of the amino acid, and one is of the IMP and others. They are actually inhibitors of the synthetic pathways, and they affect both de novo and salvage pathways. So this is the reaction of how the folate work. By reductase activities, or in this case, you can see that how the transformation of folate to finally form the FH4 molecule, because folate is a very key molecule in the all biochemical reaction as a one carbon donor molecule. So if the folate analogs are there, they can inhibit this reaction, because that's not a real molecule, and that's how, as I said, they are used in a as a control for cancer. For example, leukemia, this folic acid analogs are used as a tool to control. Okay, now we are going towards the next section of the nucleotide. We studied about synthesis, and we studied both the salvage and the de novo. Now we go towards degradation of the purine nucleotides. Now, in case of the degradation of the purine nucleotides, it's not that complicated as that of the de novo synthesis. So you can see here that like we are starting from the AMP. Why we are starting from the AMP? Because we know that it's ATP to ADP, and then ADP to AMP is just the loss of the phosphate group. So we have the adenosine monophosphate, and this is being acted upon by enzyme adenosine deaminase. Now the enzyme itself indicates what it is going to do. It is going to remove amino group from this. And as a result of this, the AMP is transformed into IMP. Then this IMP is further transformed into hypoxanthine. This is a basic transformation or rearrangement. Then hypoxanthine can be converted into xanthine by enzyme xanthine oxidase. Similarly, GMP can also be converted into xanthine, and then this xanthine, acted upon by the same enzyme, will be converted into uric acid. So you can see that this table, this diagram indicates you that the end product of the purine metabolism is uric acid. So uric acid is the final end product of purine nucleotides. It is excreted as an end product in mostly in primates, birds, and some other animals as well. The rate of uric acid excretion by the normal adult human is about zero point 0.6 gram per 24 hours, uh, and it can be increased partly because of ingested, or the purine that you have taken in your diet, and in part from the turnover of the purine nucleotides as well. The normal concentration of the uric acid in the serum of the adult is in the range of 3 to 7 milligram per deciliter. Now, there is a condition related to the uric acid disorder, and that condition is being commonly known as gout. What actually happens in the gout? You can see the picture here. This is sodium urate crystals. So, what is actually happening in the gout? It's a disease which is primarily is the disease of the joints, common in the males, and this is primarily caused by elevated levels of uric acid in the blood, definitely. So what will happen? The joints become inflamed, they become painful, and a bit of arthritic because of the deposition of abnormal crystals of sodium urate. So when you have got elevated level of the uric acid, that uric acid reacts with the sodium to form sodium urate. And once it is formed, they transformed into crystals, solid crystals. And these crystals will be accumulated in the joints, causing inflammation and pain. The kidneys are also affected by this because the excess uric acid is also deposited in the kidney tubules as well. So, the uric acid relationship and the gout, you can see here that as a result of the catabolism of, hypoxanthine and xanthine, you got the uric acid and the level is elevated, about 8 milligram per deciliter. So normally it goes out of the body, and that's what is the routine. But what happens that if it's not going through this and it's a much higher amount, then it's transformed into urate crystals, which is, as I said, the uric acid combined with sodium, and then it's got accumulated in the joints, and you can see that that's how it affects the joint movement and bit of arthritis condition. This is advanced gout condition, which is in a very, very severe form. Now, there are certain compounds that can be used to treat the gout, and one of the compound is allopurinol. Allopurinol is being referred as a suicide inhibitor used to treat gout. And you can see the structure of this allopurinol is pretty much uh, analog structure of hypoxanthine. So we can see here that this reaction which we studied, that guanine and hypoxanthine are converted to xanthine by the enzyme xanthine oxidase. This allopurinol is the inhibitor of this enzyme. So in this case, you can see that this allopurinol is going to block the formation of xanthine, then it will block the synthesis of urate. So actually, this allopurinol is an inhibitor of this enzyme xanthine oxidase, and that's how it stops the formation of a uric acid. And less uric acid means less chances of the crystal formation. Okay, so we have completed till now the metabolism of purine nucleotides. Just to summarize you that what we studied, we have studied about the basic intro of the nucleotides, the types, purines and pyrimidines and all that kind of things. Then we started with the synthesis of purine nucleotides, and we studied both de novo and salvage pathway. We studied about the regulation, and then we studied about the degradation of the purine nucleotides. Now in this section, we are going to start the synthesis of pyrimidine nucleotide, which is the second category of the nucleotides. And we do have the same process like we have in purines. We have a de novo synthesis in this case as well, and the concept is the same. But that is a bit shorter than purines. The reason is that in the purine, if you remember the structure, purine is formed by a six-membered and a five-membered ring. So it's a bit of a more complex structure, while pyrimidines are single ring. So they are easier in terms of, when it comes to synthesis, less steps or less complicated. Pyrimidine ring is made first, and then it's going to attach with 5 carbon sugar, while in case of the purine biosynthesis the situation was different. So in the case of pyrimidine de novo synthesis, there are only two precursors, aspartate and glutamine, plus a bicarbonate, which is definitely for their continuation carbon atom, and it results in the formation of six-membered ring. So six-membered ring is formed by aspartate, glutamine, and bicarbonate. Overall, The de novo synthesis of the pyrimidines is of the 6 steps. जबकि purine में आपने पढ़े थे, there were 11 steps. The product is UMP, which is uridine monophosphate. So let's start. Number one, element source of pyrimidine base. So element source of pyrimidine is very much clear in this picture. You can see this is the pyrimidine ring. You can see there are 4 carbons and 2 nitrogen. So you can see that these 2 carbon, carbon number 4 and carbon number 5, sorry, 3 carbon and carbon number 6, plus this nitrogen. This is being coming from aspartate, and nitrogen number 3 is coming from glutamine, and carbon number 2 is coming from bicarbonate, or you can say carbon dioxide. So this is the element source. Okay, let's start in detail how it's synthesized. Step number 1 of the pyrimidine synthesis, de novo synthesis, is synthesis of carbamyl phosphate. Now, here comes the reaction. Bicarbonate and glutamine utilizing 2 of the ATP molecules and water molecule. You can see here the 2 molecules of ATP are transformed into ADP. That means you need energy to carry out that reaction. The enzyme is CPS2. This is the step number 1 of this pathway, and you got carbamyl phosphate, and this is carbamyl phosphate synthetase 2 enzyme. So carbamyl phosphate, and you got this phosphate group attached here. This is the first step of the synthesis of pyrimidine nucleotides by de novo synthesis. In most of the cases, if you remember, the first steps are the rate determining or the regulatory steps. So carbamyl phosphate synthetase has got 2 different types. One is of mitochondrial origin and is dedicated to the urea cycle and arginine biosynthesis, which is going on in mitochondria, while the second of its type is a cytosolic enzyme, which is this one that we have studied. And this is the committed step, the regulatory step of the synthesis of pyrimidine nucleotides by de novo synthesis. The step number 2 is the synthesis of carbamyl aspartate. And now this carbamyl phosphate is acted upon by ATCase, which is aspartate transcarbamylase, and aspartate molecule comes into action. Remember, aspartate has given 3 carbon and 1 nitrogen. So you can see that this whole molecule of aspartate is here, and it's going to attach here, giving 4 carbons and 1 nitrogen to form the intermediate product that's called carbamyl aspartate. The carbamyl phosphate is an activated compound, so no energy input is needed in this step, and you see that there is no utilization of the ATPs. The step number 3 is the ring closure. The ring closure to form the pyrimidine ring. So ring closure to form a compound called dihydroorate. So there is no specific, I would say, enzymatic reaction. Rather, it's kind of a rearrangement reaction where we have enzyme dihydroorate, which removes a hydroxyl group. And as a removal of this hydroxylate group, this is the location from where, and that results in the ring. So it's kind of a rearrangement or a shuffling of the position that results in the formation of that ring structure, which is dihydroorate. Step number 4 is the oxidation of this dihydroorate to form orotate. Now it's an oxidation reaction, so you can say that we have quinine to form reduced quinine, which is, if it is oxidized, then there should be a reducing agent there as well. Dihydroorotate dehydrogenase enzyme is there, and you got a pyrimidine formed, the first of the pyrimidine formed here, which is orotate, or in other words called orotic acid. So this is actually, these are the two hydrogens is indicated here. It's going to be lost, and that results in the double bond formation here. You can see. Step number 5 is the acquisition of ribose phosphate moiety. Now, uh, first ring structure is there, which is orotate. Now the step is to get the ribose phosphate moiety get attached, and it involves two steps. The first step, you need a phosphate donor, which is PRPP again. Enzyme is orotate phosphoribosyl transferase, and you got the first of the phosphate group along with the sugar attached. Then you have the next step, which is being by OMP decarboxylase, which is the removal of the carbon dioxide. And finally, you got the molecule of UMP. So that's why we call it decarboxylation of the OMP, because uh, the fifth step is actually transferase, where the phosphate group is transferred, and the sixth step is removal of the carbon as carbon dioxide, which is decarboxylation, to form the first of the pyrimidine synthesis that takes place, which is UMP. So this is what the phosphate group and a ribose moiety get attached, and here is the decarboxylation. So this is the bigger picture of all the reactions, a summary, I would say. So starting from there, we already have started all these steps. I'm not going to go in the discussion again. That's just a summary of all these 6 steps we have studied. Now from there, what we got was UMP. Now from UMP, we'll get UTP and CTP biosynthesis, and that's pretty simple, just like from AMP to ATP and from GMP to GTP, it's pretty much the similar case, right? We got the addition of the phosphate group going on, kinases are the enzyme, and so on and so forth. Now from uh, a monophosphate, a thiamine monophosphate, to a deoxy form, because it's also the precursor for the for the DNA. So in this case, the immediate precursor of the thymidylate is DUMP, and it has to be converted into DTMP. So the formation of the DUMP either by deamination, removal of the amino group from the CMP, or by hydrolyzation of the molecule of the UDP. So the first one is the main route, like the formation of DUMP is mainly done by the deamination pathway. So you can see here that UDP is converted to DUDP, or here you got the CDP converted to CMP. From either way, they are being converted to DUMP by this process of deamination. Then this DUMP is being acted upon by enzyme DTMP synthetase, which gives you DTMP, and then DTMP will be adding on with the phosphate to form di and then triphosphate. And that's how we got the formation of DTMP, and then from DTMP to di and then tri. Thiamine monophosphate, thiamine diphosphate, and then thiamine triphosphate. DTMP synthesis at the nucleoside monophosphate level is being shown here. It's the same thing, but it's more detailed, where you can see this DUDP and DCMP can be converted into DUMP. That is being shown in the previous picture as well. And this then DUMP is being acted upon by enzyme thymidylate synthetase. There is an oxidation reduction going on and results in the formation of GTP, which is a precursor molecule for DNA synthesis. Just like in the previous case, we do have a salvage pathway for pyrimidines as well. And these are a few of the salvage reactions. Uridine cytidine, which is a precursor already available in the body, utilizes ATP enzyme uridine cytidine kinase, will give you direct pyrimidine nucleotides. Similarly, deoxythymidine can be converted to DTMP, deoxycytidine can be converted to DCMP, and uracil thymine can be converted to UMP TMP. So these are one-step reactions, and directly from these of the intermediates and the precursors or the metabolic products of the degradation can be directly converted by single step to the pyrimidine nucleotide. So that's what the salvage pathway is all about. In this case, we also have anti-metabolites. Just we have anti-metabolites of purine nucleotides, and the role was, the similar. Anti-metabolites are the ghost molecule. They look like the original molecule, but they are actually the inhibitors. So anti-metabolites of the pyrimidine nucleotides are pretty much similar with them of the purine nucleotides. Here you can see one of the pyrimidine analog is here. You have got thymine, the original molecule, and its anti-metabolite is 5-fluorouracil. Similarly, we have amino acid analog, the same one as the serine because amino acid is the same, and we have the same analog for the folic acid because that's also the same. So the only difference was in the pyrimidine analog because in case of the purine, their structure was different. But these two analog are the same, and they have the same function as we studied earlier on. They are the inhibitors, and they are the best tool to be used to treat cancer. Now, what are nucleoside analogs? Nucleosides, you know the difference of nucleotide and nucleoside. So this is a nucleoside which is without a phosphate group, and they do have analog as well. So arabinosyl cytosine is the analog of cytosine, which is again keeping in mind, I repeatedly said that these all analogs are metabolic inhibitors. So it inhibits the synthesis of DCDP. Finally, the degradation of the pyrimidine nucleotides. This is again a summary, just like we haven't gone in the detail of the degradation for the purine as well, just being shown with a flow chart, and here is also a flow chart. So you can see that cytosine is being converted to uracil, and then uracil is being further converted into oridopropanoate, and then it is finally converted into alanine with the loss of carbon dioxide. Similarly, if we go to the thymine, thymine is degraded to oroidoisobutyrate. Then by that, it's converted into you know NH3 group. So the end product of pyrimidine nucleotides is very different from what we have in purine. In purine it was uric acid, but here we have NH3 and carbon dioxide as the end product of pyrimidine degradation, and both of them are highly soluble product in highly soluble in water. So the summary of the purine biosynthesis, just to summarize, IMP gives to AMP and GMP, and then from AMP to ADP and GDP, and then the triphosphates, and from triphosphates to the deoxyforms as well, which are the part of the DNA. This is the summary of the pyrimidine biosynthesis, where you have got the UMP as the main molecule, and from UMP you got deoxyforms. Sorry, UMP converts to UDP and then to UTP, and then UTP converts to cytidine triphosphate and diphosphate. It's all interconvertible into different forms. So we have the normal form, which are in the RNA, as well as the deoxy form, which is the precursor for the DNA. So the summary of the nucleotide synthesis is purine buildup uh, on ribose, and PRPP synthetase was the key step, which was the first step for the IMP synthesis. Remember, in pyrimidine rings build, the ribose is added later on. So the first synthesis was of the UMP, and the key enzyme was CPS2, and salvage pathway is important for its synthesis. So these are a couple of points to be just recalled, that in synthesis of the purine nucleotides, de novo synthesis site characteristics and element sources, you have to look for that. In the salvage pathway, look for its definition, significance, and we discuss about the a syndrome, Leschnin syndrome. So these are the actually important points that you have to summarize and go through when you go through the presentation again. So with this, we have completed the lecture, and we have completed this first topic of metabolism of nucleotides. So I'm going to finish the class here, and I'll share the lecture with you guys once the recording is available. And in this case, you guys can ask about the questions on the WhatsApp group or in the next class, whatever at your convenience. Thank you very much. Have a great day. Khuda Hafiz.